Continuing with the discussion of prokaryotic translation stages, uh, next stage is elongation. So, the requirements for elongation stage is the functional 70S initiation complex, a pool of amino acid transfer RNAs, three elongation factor TU, TS, and G in prokaryotes, and energy in the form of GT. So, how does the elongation process continues? So, it requires the functional 70S ribosome forming the initiation complex that contains the messenger RNA precisely present such that the AUG codon is at the ribosomal P site. The initiating formallated methionine transfer RNA is present at the ribosomal P site by codon-anticodon interaction. And the next amino acid that will now enter at the ribosomal A site. So only the first initiating amino acid transfer RNA, when it enters, will bind to the P site. During the rest of the translation process, all the incoming amino acid transfer RNAs will first bind to the ribosomal A site. So we basically have the pool of amino acid transfer RNA molecules. So the next amino acid transfer RNA that is to bind to the ribosomal A site will first form a larger complex by binding with elongation factor TU GTP complex. Now, by codon-anticodon interaction, the complex between amino acid transfer RNA, elongation factor TU GTP complex will pair with the codons on the ribosomal A site. Simultaneously, there will be release of elongation factor TU and hydrolyzed form of GTP releases, GDP. So you will have release of a complex of elongation factor TU GTP. So here, the elongation factor TU-GTP complex has to be regenerated, which is done with the help of the second elongation factor, that is elongation factor TS-GTP, that exchanges its GTP with the GDP of elongation factor TU-GDP, that results in regeneration of elongation factor TU-GTP complex. Now we have the next step is peptide bond formation, wherein the amino group of the amino acid that is bound by its transfer RNA to the ribosomal A site will make a nucleophilic attack at the carbonyl carbon of the first amino acid bound to its transfer RNA. This leads to formation of a bond between the first and the second amino acid. A peptide bond is formed, and this results in formation of a dipeptidyl transfer RNA at the ribosomal A site and an empty transfer RNA at the ribosomal P site. This reaction is actually catalyzed by the 23S rRNA of the 50S ribosomes. Next step in elongation is translocation, which is catalyzed by the elongation factor G, also known as translocase. So this will also enter in a form of a complex of elongation factor G, GTP complex, which will catalyze the translocation reaction. During this reaction, the ribosomes will shift towards the 3' end of the messenger RNA molecule by a distance of 1 codon. As a result, the dipeptidyl transfer RNA, which was originally at the ribosomal A site, will now be present at the ribosomal P site, as can be seen here. And the empty transfer RNA that was originally present at the P site will now be present at the E site or the exit site. And the A site is now empty and it will allow entry of a new amino acid transfer RNA elongation factor TU GTP complex and allowing codon anticodon interaction, which will allow binding of the amino acid transfer RNA at the ribosomal A site. Again, you will have peptide bond formation and the reaction will keep continuing till you reach the termination sequence. From the E site, the empty transfer RNA simply falls off and the process of elongation will continue with the rest of the messenger RNA. So all these points that we had just discussed are written here for your reference. Coming to the last stage, that is termination. So termination is been achieved when one of the three termination codons, that is UAA, UAG, or UGA, are present at the ribosomal A site. So when any one of these three codons are present, that leads to binding of a release factor. So in case of prokaryotes, three release factors have been identified: release factor 1, release factor 2, and 3. One of the three release factors will bind to the ribosomal A site when the termination codon is present over there. Once bound, it brings about hydrolysis of the bond between the carbonyl group of the peptide chain and the transfer RNA molecule, hydrolyzing the bond, releasing the amino acid or the peptide chain. This results in a deacylated transfer RNA. As soon as the peptide chain is lost, the entire complex simply dissociates, releasing the messenger RNA, empty transfer RNA, release factors, and the 30S and the 50S subunit will again dissociate, immediately after which the initiation factors can interact with the 30S ribosomal subunit to begin a new translation cycle. That's it about prokaryotic translation process. Thank you, and kindly like and share if you like the content, and subscribe to get a direct update on the uploaded videos. Thank you.